Welcome to the problem-solving set on altered dominance relationships and multiple alleles. Question 1 asks, what form of dominance would explain the following flower crosses, with colors in regard to petal color? For A, it's a blue petal color times a yellow petal color, and all the offspring are blue. Now this is a form of complete dominance. With the blue allele being completely dominant over the yellow allele. So blue is down. And that's a characteristic of complete dominance because one of the phenotypes completely masks the other. For B, it says a blue times a yellow, and all the offspring are blue with yellow spots. Now here, both phenotypes are showing in the offspring, which is characteristic of codominance. And that's the same like AB blood groups. If you're AB, you have both sugars on your red blood cells. For C, blue times yellow, and all the offspring are green. Now this is a characteristic of incomplete dominance. And that's because the offspring phenotype is a mix somewhere in between the two parental phenotypes. And for D, it says blue times yellow, go to all yellow. Now here again it's complete dominance, but this time it's the yellow that's dominant, so this must be a different species of flower. So let's just write that down. Codominance is where both phenotypes are showing. And incomplete dominance is where the phenotype is somewhere in between the two parental. Okay, and those are the three types of dominance. Question 2. 4 people whose blood types are denoted below are in a car accident and require blood transfusions when taken to the hospital. If you work in the blood bank, what samples could you prepare for each patient? What are the possible genotypes for each patient? So let's look at Mary first. She has an AB blood type. And this is denoted as the universal acceptor. Now that's because she has both the A and B forms of the polysaccharide on her red blood cells, so she doesn't make antibodies against either A or B. So if you were working at a blood bank, you could prepare an A sample for her, a B sample, an AB sample, or a sample from an individual with O blood type. So she can get any blood transfused. Now for her genotype, because she's both A and B, that means she must be big I A and big I B. For Tom, he's blood type A, which means that he has antibodies against B, so he can receive either A blood or O blood. Now he, with blood type A, he could be either IA, or IA and little I, and that's because the big I's are always dominant to little I. For Sarah, she's blood type B. So it's the same with A, she can only get her own blood type B or O. And her genotype can be either big IB, or big IB, little I. Again, because the big I's are completely dominant over little I. Now for Mark, he's blood type O, which are the universal donors. And as you can see, each of these other blood types can receive O. However, he can only receive O. And that's because he has antibodies against both A and B. So to be a blood type O, the only genotype possible is little I, because any of the big I's would be completely dominant over that. Question 3. A woman with blood type AB marries a man with blood type A whose mother was type O. What was the man's genotype? Now he has type A, which means he could either be big I, big A, or big I, big A, little I. Now because his mother was type O, which is little I, you know that he had to get one of those little I's, so therefore the man's genotype was big IA, little I. So let's do the cross for this married couple. So we know his genotype is big IA, little I, and her genotype, because she's AB, must be big IA, big IB. So let's do the Punnett square. Okay. So you get big IA, big IA, big IA, little I, big IA, big IB, and big IB, little I. Okay. So part B, what is the probability that their first child will be a boy with blood type B? So again, these are two different events. So you have a half chance of getting a boy, and it says a boy with blood type B. So which of these offspring are blood type B? This is blood type A. This is blood type A. This is blood type AB. And this is blood type B. So out of the 4 offspring, one of them is blood type B. So that's a 1/4 chance. So the total probability for part B is 1/8. For part C, it says what are all the possible genotypes and phenotypes for their offspring. So the genotypes we could just take from the Punnett square. And this is phenotypically type A. Then you have big I, A, little I, again, type A. Big IB, little I, is type B. And big IA, big IB, is type AB. So these are all the genotypes and phenotypes of their possible offspring. Question 4. The dominance hierarchy for rabbit coat color is as follows. For the cross big C, little ch, times little ch, little c, ch, what is the probability of Himalayan progeny, and what is the probability of agouti progeny? So let's do the Punnett square for this cross. One parent has a big C and a little c, h, and the other parent has a little c, h, and a little c, ch. So let's just fill out the Punnett square. Okay, so these are the possible genotypes of the progeny. Let's do phenotypes. So we know that big C, or full color agouti, is dominant over the other 3 alleles. So that means that this individual and this individual are both agouti. So that means that 1/2 of the progeny are agouti. Now this individual is little ch, little ch, so they are Himalayan. So 1/4 Himalayan. And this individual here is little c, ch, little ch. So chinchilla, which is little c, ch, is dominant over Himalayan. So therefore this individual is chinchilla. So 1/4 chinchilla. So part A was asking the probability of Himalayan and agouti. So for Himalayan, it's 1/4. And for agouti, it's 1/2. Now part B is a different cross, so let's do the Punnett square for that. It is big C, little c. Big C, little c. And little ch, little c, ch. Little ch, little c, ch. And then just go across and fill that out. Okay, so these are the genotypes. Again, let's do phenotypes. Remember that big C, or full color agouti, is dominant over the other 3 alleles. So that means that these 2 individuals are agouti. So 1/2 full color agouti. Now both the Himalayan and the chinchilla are both dominant over little c for albino. So that means that this this individual here is definitely Himalayan, so it's 1/4 Himalayan. And this individual here is definitely chinchilla, so 1/4 chinchilla. So the question's asking, what is the probability for chinchilla progeny and what is the probability for albino? So for chinchilla, it's 1/4, but for albino, it's 0. 0 albino. And remember, to be albino, you have to be both little c, little c. So even though, if you look, both of these crosses came to the same progeny. 1/2 agouti, 1/4 Himalayan, and 1/4 chinchilla, even though both crosses were slightly different. So for when there's multiple alleles, it's your best bet is to do a Punnett square and keep in mind the dominance hierarchy in order to see the phenotypes. Question 5. Milkweed flowers have 2 alleles for petal color that are incompletely dominant. One allele is for red flowers and the other is for yellow flowers. Heterozygous plants produce orange flowers. Determine the ratio of flower colors for the offspring of the following crosses. So before we do the crosses, let's define our alleles and the phenotypes that go along with them. So let's use the allele C for color. So we have C1, we'll say equals red. So in order to be red, you have to be C1, C1. Now for yellow, let's say C2, C2 equals yellow. And because these alleles are incompletely dominant to one another, the heterozygous C1, C2 is the orange color, which is a phenotype somewhere in between the two parentals. So now let's do these crosses. For A, we have a yellow flower times an orange flower. So again, to be yellow, you have to be C2, C2, and to be orange, you have to be C1, C2. So if you fill that across, the ratio here, half of them are C1, C2, which is orange, and the other half are dominant, homozygous dominant C2, C2, so half are yellow. Okay. For B, you have a red flower plant times a red flower plant. So again, to be red, you have to be C1, C1. And as they're both C1, C1, you can tell that 100% of the offspring are going to be homozygous C1, C1. So it's 100% red flowered offspring. Okay. For C, you have an orange plant times an orange plant, which are the heterozygotes. So you have C1, C2, C1, C2. And this is similar to a monohybrid cross, but because of the incomplete dominance, you'll see that the ratios are a little bit different. So you have C1, 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 C2, C1, C2 again, and C2, C2. So you have 1/4 is C1, C1, which again is red. 1/4 is C2, C2, which again is yellow. And then the two heterozygotes of the offspring, C1, C2, are orange. So you get 1/2 are orange. Okay, so it's pretty simple, but let's keep going. So for D, you have a red plant times a yellow plant. So again, to be red, it's C1, C1. And to be yellow, it's C2, C2. So all of the offspring here are going to be C1, C2, which is the heterozygous, which means it's 100% orange. For E, you have a yellow plant times a yellow plant. And to be yellow, you're C2, C2. And again here, everyone's going to be C2, C2, which is 100% yellow offspring. And for E, finally, you have a red times orange. So to be red, you have to be C1, C1, and the orange is heterozygous, so you have C1, C2. So you have half being C1, which is red, and the other half being heterozygous C1, which is orange. Question 6. Blood tests can detect different antigens, 4 of which are described below. You have the ABO type, which we've been familiar with, where big IA and big IB are codominant, and little I is recessive. You have an RH type, where RH plus is dominant to RH minus. You have the MN type, where M and N are codominant alleles. And you have the XGA type, which XGA plus is dominant to XGA minus, and this is an X-linked allele. So a mother is trying to determine the father of her daughter. Based on the lab results shown below, which man could be the real father? So let's go through each of the 4 antigens one by one and see if we can cross anybody out. So let's first look at the ABO blood type, which is here. The daughter is AB, which means she has to be big IA, big IB. Her mother is blood type A, which means she could be big IA, or big IA, little I. So this daughter could definitely get this big IA allele from her mother, either way. And that means that she has to get this big IB allele from her father. So man number 1 has a B blood type, which means he could definitely pass on the big IB allele. Man number 2 has an AB blood type, which means he also has a big IB allele that he can hand off to his daughter. Man number 3, however, is blood type O, which means he's little I, and therefore doesn't have this big IB allele to give to his daughter. Therefore, man number 3 cannot be the father due to the ABO blood type, so we'll cross him out. Okay, now let's look at the RH factor, which is here. The mother is Rh minus, and again, because Rh minus is the recessive condition, that means she must be little r, let's call it. The daughter is Rh plus, which means she is big R, little r, and we know that she's heterozygous because she had to get the little r from her mother. So that means that her big R had to come from her father. So the father must have been at least one big R. He could have been heterozygous or homozygous. But that means that the father had to be Rh plus. So let's see who is Rh plus. Again, man number 1 and man number 2 are both Rh plus, so they could possibly be the father. And just another thing to prove that man number 3 is not the father, he's Rh minus, which means he wouldn't have this big R allele to give to the daughter. Okay, now let's look at MN type, which is over here. Okay, so the mother is M, and the daughter is MN. So that means the mother has the genotype MM, because M and N are codominant, as it says up there. So if she had the N, it would be showing. So therefore, she got the M allele from her mother. That means that she had to get the N allele from her father. So her father must be MN or just N. So let's see who we can cross out now. So man number 1 is just M, which means that he wouldn't have the N allele to pass on to the daughter, who's MN. So for this reason, man number 1 is crossed out. And let's see, man number 2 is N, so he has this N allele that he could pass to his daughter. It would actually be coming from here. So on these first 3, the ABO, the RH, and the MN types, man number 2 seems to be the father. But let's just double check with the XG type. Okay, so for the XG type, XG A plus is dominant to A minus, and this is an X-linked trait. Okay, so the mother here is XG A plus, and the daughter is XG A minus. Now again, A plus is dominant to A minus, and it's X-linked. So this mother, let's call it X plus. She had to have at least one X plus. And this daughter here is A minus. So because the A minus is the recessive phenotype, she must be X minus, X minus. That means that this mother, the other allele, must be X minus because she gave one to her daughter. And the father must also give X minus. Now because males are hemizygous for the X chromosome, that means that he is X minus Y, which means then that he would be XG A minus. Even though it's the recessive phenotype and he only has one chromosome, males are hemizygous, so any chromosome allele that they have, that would be their phenotype. So the man must be XG A minus. And for man number 2, he is that. So through all 4 antigen tests, we'd say that man number 2 is the father. Question 7. The dominance hierarchy for lentil seed copattern is marbled is dominant to dotted, which is codominant with spotted, and everything else is dominant to clear. A marbled lentil is crossed with a spotted and dotted lentil. If the marbled lentil were homozygous, what ratio of offspring seed patterns would be expected? So let's write out that cross. So we have a marbled lentil that is homozygous, so it is C big M, C big M, and it's crossed with a spotted and dotted lentil, which is C D, C S. And remember, they're codominant, so both the spots and the dots phenotype will show. So let's do the Punnett square for that cross. Okay. Oops. Okay. So if you see, every offspring from this cross has at least one CM. Now because CM is marbled, is dominant over all the other alleles, all these offspring, they're 100% marbled. Okay, so for part B, it says if the actual offspring ratio is 2 marbled to 1 dotted to 1 spotted and dotted, what was the genotype of the original marbled lentil? So let's write out the genotypes for these offspring phenotypes. It says 2 marbled. Now again, because marbled is the most dominant allele, they would only need one big CM. And the other one could be a dash because we're not sure what it could be yet. For a dotted, let's see, it is CD, and it could either be CD, CD, or CD little c, because dotted is dominant to clear. And for spotted and dotted, that genotype definitely has to be CD, CS, because they're codominant alleles. And remember, for every gene locus, every individual can only have 2 alleles. So again, the parents are marbled, it's one parent, and the other parent is spotted and dotted. So we know that this parental genotype is CD, CS, and this one is at least one CM. So out of these parental genotypes, what other allele here could give these phenotypic patterns? So let's see. To get spotted and dotted, one of the CS could come from this parent, or the parent could give their CD. However, to get dotted, this dotted came from this parent, but where did this one come from? It would have to come from this parent. So right now we're just going to assume that it's CM, CD. And if that was the case, then this spotted and dotted would also work, because the CS could come from the spotted and dotted parent, where the CD could come from the CM parent. So let's do a Punnett square just to be sure we get the right ratios. If this is our guess for parental. And again, this would look marbled because it has the one CM allele. So if we do the Punnett square, you have CM, CD, and CD, CS. Okay, so these two, you get 1/2 or 2 marbled. CD, CD gives dotted. And CSCD gives 1 spotted dotted. So that matches the phenotypic ratios we were given in the problem. So the answer here, that original marbled parent, was CM, CD.